continue. Recording. Jazz hands. So all of this material is collected from not the internet, but the presentation you have access to. Record it. There's lecture 13 is the most important lecture for this paper. Then also the most important files for this paper are the Lab 6 handout about eye color. And the, there's another Word document. The UPD what to put in your lab report thing. You know what? I'm actually going to change this share a little bit, and I'm going to share just my whole desktop so you can see what a nightmare my life is right now. So this file here, format for lab write-ups, correct spelling, grammar count, and this is an important, like all of these things. So pay attention to that. And that's available on Canvas in the module about this paper. This is a Lab 6 fly eye thing. This is also so darn important. These questions need to be answered in your paper. And also important from that module is this rubric. I can't emphasize enough that you should be going over this rubric regularly as you write. And at the end, make sure you can check off all of these boxes. You can download this rubric yourself from the module. This is how we will be grading you. So make sure that you grade yourself well before giving us the paper. Sound good? Okay, so let's talk about fly eyes. Not you, now. So many things. There we go. Okay. From the top. This thing is in the Fly Eye Lecture 13 eye color thing. This first slide, slide 2. And it looks like a chaotic thing. I'm just breaking it down a little bit here. There are 2 parallel pathways going on. One regulates orange and yellow pigments, and the other regulates brown pigments. So for this pathway here, and they happen in parallel. So these proteins don't directly regulate each other and interact, but they maintain the same, but they affect the same phenotype, eye color. So they blend essentially. So if something is broken in the pathway to make orange, you can't make orange. And that's what a mutation is. A mutation is just broken genes. So if, say, the brown gene, which is a transport protein, this guy here, if brown is broken, that means that you can't make orange pigment. So you're going to have brown eyes because all that's left is this pathway. And so the same works here if, say, scarlet or vermilion or cinnabar, all of which have phenotypes that are very, very similar to each other, are broken, just one of them is broken, you're not going to be able to make brown. So you're just going to have orange eyes. And that's an important thing to keep in mind. These 3 genes, scarlet, vermilion, and cinnabar, are in a linear pathway together. So if one of them breaks, the whole system doesn't work. And if 2 of them break, the whole system doesn't work. And it doesn't matter which one breaks, it'll all look the same. You'll have orange eyes, you won't be able to make brown. Okay, so take home message, when you break a gene necessary to make brown, you will get orange eyes. Not because your parent passed you down the orange phenotype, but because your parent failed to pass down the ability to make brown. This is the example that she has given us for the chromatography. She wants us to use this image in the paper. All of these three, biopterin, sepiopterin, and drossopterin, are these things: biopterin, sepiopterin, drossopterin, and they're all in the orange pathway. So if you have a mutation that breaks brown, you're going to have orange eyes, and you're going to have all of these pigments functioning. And because the pigments are made in the eye after the various different bits are transported there, if you can't transport the thing there, so either you lack white or you lack brown, you can't transport the thing there, then you're not going to make the pigments. They won't be present. So here we see in this brown mutant, so a brown phenotype. So we've got the gene brown broken, likely, for the brown flies that we looked at. And you see that there are none of those orange pigments. They can't make the orange pigments because they can't get the ingredients to the place where they would make them. And that is because this transport step is broken. And of course, just looking at this now, we've spoiled one of the questions, right? Because we've been trying so hard to keep from you whether or not it's one gene or two. But here we can already see we've got an orange phenotype and a brown phenotype. Because this is brown, that means that these flies with brown eyes, they can still make the brown pigment, which means that they have a functional white gene. They must, because the ingredients to make brown have gotten to the eye. Which brings us to this question from the Fly Eye PowerPoint. Now we're on slide 4. And we can see that these two options, either you're mutant for white or you're mutant for brown and something else, we know that they're not mutant for white because we found these flies. We were working with these flies. Because these flies exist, we can already rule out the one gene hypothesis. Are you with me? Feel free to speak up in the chat if you are lost. Amazing. We're going to move on now to this beautiful phenomenon here. So I've made you a pedigree because I care about you. You can make this pedigree on your own if you want. You have the power. But I've just gone ahead and made it. These are just the words from this file, these words translated into a pedigree. That's what this is. And then these numbers are Thursday's lab numbers. The numbers in the boxes. So the questions we need to answer are, how many genes? We've already answered that. Because we know that we have browns kicking around, it can't be white. So in order to get white eyes, two things need to be wrong. We need to fail to make brown, and we need to fail to make orange. So because we have the brown eye phenotype available, we know that it's possible to only be able to make one of them. So we know that brown is broken. And she spelled that out for us, right? The single gene option is that it's mutant for white. But the 2 gene option means that it's definitely mutant for brown, which is on chromosome 2. Plus, it's also mutant for a second gene, one of these 3 options. And these vermilion, cinnabar, and scarlet are these guys, scarlet, vermilion, cinnabar. All of those parallel genes that are the different components of the pathway. Scarlet is required to transport the bits to the eye, and vermilion and cinnabar are necessary to synthesize the pigment.
So just to reiterate again, so
Yeah.
the fact that the reason why we can rule out the single gene is because we found brown eye flies.
Yeah, that would also be true if we found orange eye flies, right? If you can separate them out so that we see white, but we also see some pigment, but not all the pigment, but not like one pigment, but not the other pigment sets, then you know, because they're parallel pathways, you know that they have to be 2 things, there has to be 2 things going wrong.
To induce white eyes.
To induce white eyes. You have to knock out both pathways individually because you can bring them online individually by rescuing just one. And that's emphasized here too, right? So these are white, that means that they're double recessive. AABB. Because the offspring are wild type, but all of them, we know for sure that their parent has to be homozygous dominant because the offspring need to be able to get reliably every single time, get a dominant allele from their other parent. So these guys are both going to be heterozygous. And that helps us with determining which of these, with ruling out one more of these. Does that make sense? So here we know that brown is on chromosome 2, which is an autosome. Do we see any sex-linked traits? No. We don't see any sex-linked phenotypes going on here. And most importantly, this cross P produced exclusively wild-type phenotypes. But if cross P, if this female had an affected X chromosome, then her sons would only get her X. And if that X was where our mutation was, they would fail to receive a functional version of that gene because she doesn't have any to offer. So these are heterozygotes, these F1s. The red ones are. The phenotype is heterozygous. And the reason for that is that they're getting 1A from each parent.
Yep.
They're getting 1B from each parent. So necessarily, they must have one big and one little for each. There's no other way to do that. So when we get to cross A, B, and C, this is cross A. We've got a heterozygous F1 and a mutant, which means it's homozygous for white. So it's got to be AABB. And we already decided this was AABB. Does that make sense? So because this is a heterozygous, this is a test cross. And she gave us that information here. It's a test cross on the Lab 6 handout. For this test cross, we're testing this genotype. We're trying to figure out whether this is a double heterozygote. And we're getting these results. So I'm not going to spell out what these results mean. But if you're a little confused about how these results mean, I want you to go look up dihybrid cross. Di to hybrid means different. In this case, it's mutant. Cross. So when you look up dihybrid crosses, and I think that I've posted stuff about that previously, but there's also stuff about that on my YouTube page. There's stuff about that on Kevin's 202 YouTube page that's linked on my useful files page at the top. So if you're struggling with why we have these new phenotypes and the way that these alleles are segregating, go there and go refresh yourself. I know for some people 202 was a long way away, so that's real. But as you're looking at the segregation patterns, remember that these things map onto these candidate genes. Okay, so we've already decided this is autosomal, yeah? So let's come to here. That means we can already rule out vermilion. Right? I guess let's not do that color. Let's do light gray. There we go. So we can rule out vermilion because it's on X. Now brown, which we're definitely counting, is on chromosome 2. No, not that color. Let's make it purple. Nah, we already have purple. Let's do sure, why not? Chromosome 2. So cinnabar is also on chromosome 2. So that notation is slightly different. You see, there's no semicolon here between the CN and the BW. That means that they're on the same chromosome, they're linked genes. Scarlet is on the 3rd chromosome. So there's a semicolon between them, they're unlinked genes. So this next question is, hold on, let's just change this titch. Linked or unlinked. Okay. So how many genes? There's 2. It's autosomal. Linked or unlinked? Here's our next question. Linked or unlinked? Because that determines whether it's cinnabar or scarlet. Those are our options. So if it's linked, hello. No, it's fine. Apparently I'm recording. It's very exciting. If it's linked, if we have 2 linked genes, then what's going to be different versus if it's unlinked? Anyone can volunteer. And type it in the chat. What will we see in these crosses? Aha, we've got a chat. The ratios, for sure. Thank you, Anna. So there'll be different ratios if they're linked versus unlinked. And this has to do with the dihybrid test cross. This is what we're testing. And specifically, we're going to compare cross A to cross B. Because cross A, we used our heterozygous hermaphrodites. Or sorry, heterozygous females, not worms, flies, not worms, flies. We're going to use our heterozygous females. This file is on the is commented on the announcement. So these are heterozygous males, and the cool thing about flies in males versus females, what's the difference? What can't happen?
Males have recombination.
Males cannot recombine. So we can see at a glance here, cross B has heterozygous males, and it's making heterozygous gametes. And that meiosis process is not resulting in crossovers because it's male. And so these genes are not independently assorting in those gametes, in those sperm from this male, because the males don't recombine. If it was assorting independently in this cross, we would know they're not linked, because it wouldn't matter if they can recombine, they're not on the same chromosome. In cross A, we see that because these females are heterozygous, they're making heterozygous gametes. They're going through meiosis and they're recombining. So we get a variety of phenotypes. And we're seeing the orange and the brown. We're seeing these single mutants, as opposed to just the white double mutants. Are there any questions about how to work that out? You good? We good? Online? Everybody happy? Okay. So that's the answer to this question. They're linked. They're linked because we see recombination here, and we do not see independent assortment here. Which means we can map them. Because I'm not going to map them. You can't make me. That's math. I don't want to deal with that. So y'all will have to go off somewhere else and map. But there's a PowerPoint presentation on my page that will help you do that. Because I once mapped. I did that for like 2 years at the beginning of my PhD. I'll never do it again. You can't make me. Okay. So this is an important comment that I wanted to make. This is how we worked out each of these questions. We asked, what information do I need? And how can I get it? And then we asked, what would I expect to see in these scenarios? And what do I see given our results? So this is also a structuring thing when you start writing your paper. At the beginning of your results section, you're going to organize your results. Here's how we, here are the tools that we made. And each result kind of builds on the previous result because you're assembling your tool set. And you're saying, okay, here are some facts. We're lining them up in a row. And then you turn to your discussion. And you say, okay. And you say, okay, so now that we've got all these tools and we all understand what we're talking about, what do they mean? How do they interact? What do they say about the biology of these worms? So this is the last 2 slides that I prepared here. We've got some new sections that are different from the first paper. The abstract always comes first in everybody's head. Write the abstract last. Don't write it first. Because if you try to write it first, you're going to feel overwhelmed. You're not going to have a good idea of what's going into the paper to begin with. If you write it at the end, it's actually really simple. What an abstract is, it's all of the topic sentences of all of the paragraphs from your introduction, results, and discussion assembled into a paragraph and then edited for clarity. And I recommend when you're doing your research for this paper and for the background to look at some examples of abstracts and see how that plays out. because it really is just an outline truncated into 200 words. I recommend writing your methods first. It gives you a good footing for what you did. And also, you're assuming that the reader has read the methods first. So when you go to write your results in discussion, you'll know what the reader knows. And as you're writing those, if you come across something you haven't mentioned in your methods, you can go back and add that in. Results in discussion are sometimes hard to separate out. I struggle with it because when I write a result, I want to then write a sentence about also it's really cool. Also, it means this thing. And you have to hold back. The results are, we did these crosses and got these phenotypic ratios as illustrated in table X. The discussion is, cross A is either a dihybrid cross or a monohybrid cross. If it were dihybrid, we'd see this. If it were monohybrid, we'd see this. We saw this, therefore, we conclude this. So all of that analysis is the discussion. And the discussion is longer, and it's weighted more heavily. See, results are 5 points. The discussion is 14 points. So, and if you put all of your discussion in your results, you're going to lose points, lots of them. The discussion is where you run your chi-squared test. The discussion is where you discuss how the chromatography results tie into the cross. Sound good? And for the love of God, follow instructions. I cannot emphasize this enough. This UPD, what to put in your lab report doc. This thing here, follow it religiously. Don't leave anything out. Make sure every single aspect is accounted for. There are even points, bullet points in the discussion. It's very specific what we want. And then you can see that reflected in the rubric. It's very specific what we're looking for. And in the references, we have these examples. Do it like this. Exactly like this. 0 exceptions. Okay, where was I? Okay, read this when you get overwhelmed. If you're writing, and as you're writing, you're like, oh, there are so many things I want to say, or I don't even know what to say next. I'm completely at a loss. Go to this paper, go to this page, open it up, find where you are, and follow instructions. That's one of the lovely things about this class is that we do give you very specific instructions. And writing an academic paper is not like writing a creative art paper. It's not even like writing an essay for a liberal arts class. Like this paper is a puzzle that you're fitting all the pieces together. And there's a structure and a format. Follow that. Point mining. Everybody wants to have a guide for how do I get each point? In this class, you have a guide for that. Actually, every single point is accounted for here. It's available. You have a guide. Don't miss out on formatting points. Follow the instructions exactly. When in doubt, be boring and right, as opposed to pretty and wrong, or even pretty and confusing. When in doubt, look it up. Don't just talk out of your butt. Make sure that you're right. If you're not sure whether to put a heading in, the answer is put a heading in. We're not going to dox you for not adding extra headings, but I will lose what you're talking about and not know what section you're in if you don't put a heading. And that's gonna hit you for readability. Don't quote the lectures. That's plagiarism. A lot of people did it. We were very lenient for the first paper. We will not be lenient for the second paper. Do not quote the lectures. Don't even put it in quotes. Don't do it. Rephrase. And that's important, not just for plagiarism reasons. That's important because I know you don't understand it if you're just quoting it. I know, I can tell. So explain it. Show me that you understand. And at the end, go through the rubric and make sure you can check off all of the boxes yourself. Remember, you're writing this to communicate to me that you know what you're talking about. And I am not a mind reader. Some students genuinely don't know whatever you think is self-evident. When in doubt, spell it out. I have a lot of these sayings, don't I? They just keep popping up. Okay. That's what I have for you. Any questions? Okay. Yeah, please.
So can we discuss like again the mechanism of how we determine it's not segment again? So you said
The mechanism what?
behind why it's not segment again.
Yes, we can, but I'm going to stop the recording,
Okay.
if that makes sense. Okay, stop recording. Y'all are wonderful. And I hope that helps.